Okay, let's go ahead and get started. On Monday, as you might recall, we focused on population dynamics, right, so how population size changes as a function of time for just an isolated population. And yesterday, we tried to extend that to consider what happens when there are different types in the population, say subpopulations that may be interacting in some way. Right, so we were applying some of these ideas from evolutionary game theory to try to understand how a population might be evolving or changing over time. And then today what we'd like to do is try to think about interspecies interactions. So when you have two or more species that are interacting, competing, maybe cooperating, but interacting in some way, what are the typical outcomes, and what can we do in terms of predicting what happens in complex communities based on what we know about the simple parts of the community. And in particular, one of the primary things that my group has focused on over the last couple of years is to try to understand, if I tell you what the pairwise competitive outcomes are, what does that allow you to say about more complex communities, say trios, quartets, and so forth? But what we're going to do is basically start on the board. We're going to focus on this Lotka-Volterra competition model, which is different from the Lotka-Volterra predator-prey model, so be aware. And then after we kind of understand this, we'll move on to the PowerPoint where we can look at some data and experimental results. All right, so when we started this whole thing on Monday, we were focused on how the population size might change as a function of time. Right, so the standard model that we often were using was this logistic model. It looked like this. All right, and we spent time thinking about what happens in this context. All right, so this is just for a single species logistic. Right? All right, now what we want to do is extend this to think about, all right, what's the simplest way of capturing the dynamics of two species interacting in some way? And we'll focus mostly on competitive interactions. All right, so now we want to go to the two species situation. So now we're going to have a dn1 dt that has its own growth rate R1. And it will still inhibit itself in a similar way. But now we want to incorporate some way in which the other species, the second species, may be influencing the first species. And perhaps the simplest way of doing this is adding some term here. And I guess this is always a question of definition. I want to make sure that I do this in an appropriate way. Yes, I think we'll stick with positive. All right, and then the same thing for the second species. Okay, sorry, and this should be N2, and here's N1 over K2. All right, so the idea here is that the first species will inhibit itself in some way by using up resources or doing something else. And the way in which the second species is influencing the first is just that it's contributing to inhibiting the growth of species 1 in the same way somehow as species 1 enters here. And so what we'd like to do is try to understand, well, what are the outcomes in this model? And do those outcomes bear any resemblance to what you observe in observational or experimental studies? So in general, when given a model like this, it's always good to just know where the isoclines are, right, the locations where these derivatives are equal to 0. All right, so for example, if we want to know about dn1 dt equal to 0. Right, well, there are a couple different ways that this can happen. All right, so of course, n1 equal to 0 works. All right, but we'll mostly be focusing on the other one, where the other way in which we can get dn1 dt equal to 0, and that's if this term is equal to 0. Yeah? Yes? You're wondering about this carrying capacity, and how it's going to depend on various experimental factors. Is that?
In that case, K1 would be, in both equations, it will be K1 instead of K1, K2. What do you say about that?
So indeed we are imagining two species occupying a given region, but the carrying capacities don't have to be the same. Right? And that could be for many different reasons. It could be that the organisms are just different sizes. Right? So then if you can support more rabbits than you can goats in a given environment. Right? So that's one way they could be different, but also they just may have different resources that they're going to be using in that environment.
So in that case, I'm sorry to interrupt you. So in that case, we would be adding, K1 plus K2 in both the cases.
K1 plus K2. I'm not sure if I understand. Right. So the K1 and the K2 in principle are just telling us if species 1 were by itself, where would species 1 go to at its carrying capacity? And here, K2 is telling us about if species 2 were by itself. And in general, there's nothing special about K1 plus K2.
I could see it as a migratory kind of condition. Say, for example, K1 is the space which is occupied by N1.
Yep.
And then it is, some N2 species comes from different place, from K2, and then it affects the, the growth rate and...
Well, okay, so you're imagining different regions now, or?
No, since you have included a competition, so how can you include a competition in a few species, in one nation? So you may, both of them are simultaneously in the same space?
Yeah, so we're assuming that here, this is a model of a well-mixed community where they're competing in the same location. So what we're now asking about is the number of individuals of species 1 and the number of individuals of species 2 within that environment. So you can imagine this as being an island where I'm telling you how many of species 1 and species 2 are present, but then we're trying to understand what's going to be the outcome of that interaction or that competition. Will those two species coexist? Maybe one species will drive the other one extinct. Or maybe it depends upon the initial abundances. So this is, of course, one question is what happens in any real system, and another is what happens in this model, and so we'd like to understand each of those. And all right, so to get to that, what we want to do is to first understand these isoclines. All right, so one condition is here. The other one is going to be the following. K1 is equal to N1 plus beta 1, 2, N2. Okay. All right, so the isocline associated with species 1 is going to be a line in this N1, N2 space. Okay. We'll go ahead and add it here. All right, so it's number of individuals 1, number of individuals 2, and we can just figure this out, right? So if species 1 is equal to 0, say, then it's going to intersect at K1 divided by beta 1, 2. If species 2 is equal to 0, then we just get at K1. Right, and that one makes sense, because if species 2 is equal to 0, then the size of population 1, where population 1 is no longer going to change, should be that carrying capacity, because this is just logistic growth again. Right? So indeed, what you'd expect is that along this axis, it should look like that. Right? Right, and we have some line that connects these two. This line corresponding to dn1 dt is equal to 0. And we can do, of course, the same thing for species 2. Right, there's one of them that is always the same here. You get this. All right. Now, of course, we want to go and we want to plot this isocline. And we're going to do that with a dashed line. And it is indeed going to be another line. All right. What we want to know is, all right, where to plot it here. And in particular, maybe we might ask, is the dashed line going to start up here, above, below, or at the same location, or depends? All right, so we're going to do a vote because we love to vote. All right, so I want to know dashed line for this isocline for species 2. Is it going to start out above, below, equal, or you can wave around if you're not, if you want to do other. Understand the question? What's that? Well, all right, so this is, you can vote in some way that, you feel is appropriate given. All right. Which one? Okay, right, so you want more information, which is appropriate. All right, so let's say that you don't yet know those things, and you can vote appropriately. All right? Okay, ready? 3, 2, 1, go. All right, so we should have some. All right, yeah, indeed, yeah, we don't yet know. All right, that because it depends on the betas and the k's and so forth. All right, and that's going to be true over here. We don't know if it's going to be above or below. It's also going to be true over here. Okay, basically 2 choices here. 2 choices over there, so there are going to be 4 qualitatively different outcomes that can happen. Okay? So what we're going to do is we're going to draw each of them, and then we're going to figure out which one is which. Okay? All right, so I'm going to... all right, so here I'm going to add a... That's one possibility. Here's another possibility. That's a line, hopefully. All right, so now we want one that goes. All right, so all of these axes are supposed to be the same. Again, as you can start staring at these and figure out what you think is going to happen in each case while I label the axes. I don't want to be accused of not labeling things. All right, and in each case, solid line is the isocline for species 1, dashed line is the isocline for species 2. And in all of these cases, what we're going to assume is that the growth rates here, R1 and R2, are both positive. And I'll label one of them over here as well. So dashed line, this should be K2. And where it intersects is K2 over beta 2, 1. Did I label that all correct? Let me. Looks okay. Yeah. All right, and our options here are always going to be the following. So this is one is for 1 wins. 2 wins, 1 and 2 coexist, 1, 2 are bistable. And I'll tell you that these are going to be the 4 things that are going to happen. And it's not, I think, a coincidence that these are also the 4 outcomes that naturally arose in the context of those evolutionary games that we talked about yesterday. But here again, we're looking at both population size of 1 as well as the population size of 2. All right, so in principle, here this is a 2-dimensional problem, whereas before, in some sense, it was only a 1-dimensional problem, but we still get the same 4 basic outcomes. All right, so let's see which one we want to start with. Well, maybe we'll even first make a note of, all right, so there's going to be some trajectories on these planes. Right? And we first want to, it's nice to think about what is going to be the orientation of the trajectories when they cross each of these lines. So let's think about this line here. All right, so the trajectories, yeah, question? No. So coexistence means that at equilibrium, both species 1 and species 2 have abundances that are greater than 0. All right, so they're both, in that sense, they're coexisting, but not, doesn't have to be 50-50. It could be 99%, 1%, right? But the idea of coexistence is that they're both surviving at equilibrium. Yeah, so if you do a deterministic analysis, then the two coexist forever. If you include demographic stochasticity, then eventually once or both, well, eventually everything goes extinct, right? But yeah, so from a deterministic analysis, they'll coexist. Whereas bistability is the situation where at equilibrium, only one or the other survives, but one survives depends upon the initial abundances of the species. All right, so what we're going to do is think about, all right, the trajectories are going to cross this line, probably, at some point. What we want to know is what's going to be the angle of the trajectories. All right, so here what we're going to do is we're going to use our arms, right, to draw the angle of the trajectories as you cross that line. Do you understand the question? No? All right, so that, you know, I mean, if you start somewhere on, all right, I'm going to go off screen. You start somewhere, N1, N2, over time, that does something. Maybe it's not going to be so crazy, but I want to know, in terms of the trajectories of the species abundances as a function of time, if they cross this line, at what angle are they going to be crossing it? And we'll, if this isn't making sense, we're going to be drawing them, and you know, so hopefully it'll make more sense. It's good to just think about it. What does this line mean? I guess is, but don't tell us the answer. If it's fine to ask. Okay, yes, question. Yeah. It's not intuitive. You're saying, yeah, yeah. So that's and so part of what we're trying to do is to gain intuition about moving on this N1, N2 plane. But you can imagine that if I gave you a time series, then we should be able to do it, right? So, for example, let's imagine, all right, I'm going to draw just down here. Let's imagine that we had an oscillation. All right, it turns out that this model does not yield any oscillations. Right, the Lotka-Volterra predator-prey model does, but this one does not, so don't be aware. But I'm going to draw an oscillation. Let's say here's N1 and N2 as a function of time, where let's say I tell you, okay, here's some nice oscillations, right? So let's say that I told you this was a prey oscillation. All right, where should the predator oscillation be? In a predator-prey oscillation, which one leads? Which one peaks first? Prey peaks and then predator, right? So if this is prey, then what we might do is say, here's a predator, right? Okay. So this is a trajectory of prey, which we might call N1, and a predator that we might call N2. Right? And this is both as a function of time, right? And then in the N1, N2 space, we might then want to understand what that looks like, right? Okay. And for example, here, N1 is high and N2 is maybe a bit lower. So N1 is high, N2 is kind of here. And as we move forward in time, now N1 is decreasing, but N2 is increasing. So we're kind of coming up here. right? Once we get to this location, now the predator population has peaked. The prey has come down kind of halfway. As we continue on, now the predator is coming down. The prey is continuing to come down. So predator, prey, they're both coming down until we get over here. Now the prey population has reached the minimum, right? So we go around, and what you see is that as you track this predator-prey oscillation as a function of time, you go around some loop in this N1, N2 space. So this would be an example of a trajectory going from a time series to the N1, N2 space. Okay. Now, the trajectories here are actually going to look rather different. And so then you get different trajectories in the N1, N2 space. But partly, I guess what we want to do is try to think about what it is that these isoclines mean in terms of trying to get it. And it's fine. If you've never seen this before, then now is a good time to start. But you're not going to be able to answer the questions necessarily. But then we'll discuss what it means. But I just want to see if how much people are understanding what these isoclines mean in terms of the trajectories. Okay, so question is, crossing this line, the solid line, what's going to be the angle of the trajectories as they're going in the N1, N2 space? So let's go ahead and vote, and I just want to see kind of where we are. Right? Ready? 3, 2, 1. All right. Okay. So the solid line we're wondering about. Okay. So what's the definition of the solid line? Yeah, the definition of it. This is what the line. That's how we got the line. Is that we asked, well, what is the conditions for dn1 dt equal to 0? Yeah, that means the population of n1 is fixed over time, and that's a trajectory along this line. So that means that when you're right here, okay, n1 and n2, in principle, are changing in a dynamical way based on these equations. But the meaning of that line is that if you are on this line, n1 is not changing at this particular moment in time. Okay. Of course, if you go forward in time, eventually n1 is going to change. Go backward in time, it may be changing, but at that particular moment, given you're at this n1 and that n2, n1 is not changing. So what does that mean about the trajectory? It actually means it's actually confusing, but yeah, so it's actually, in this case, it means it's vertical. Because here's n1. If n1's not changing, that means that you have vertical lines. So there are going to be a bunch of possible trajectories crossing that line, okay, but they're all vertical. We don't actually right now know whether they're pointing up or pointing down, but we know they have to be vertical. Okay. All right, so yeah, now we'll check. Okay, now dashed line, what's going to be the orientation of the trajectories around there? Horizontal. Okay, so again, we don't know which direction, but what we know is that they should be kind of like this. Okay. All right, what do we know about the origin? Is the origin a fixed point? We're going to vote. Is the origin a fixed point of the dynamics? Yes or no? Okay, ready? 3, 2, 1. It actually is a fixed point. All right, are all fixed points stable? No, right? So indeed, it's a fixed point. We can see that here because if n1 and n2 are equal to 0, dn1, dn2 are equal to 0. Okay, so it's a fixed point, but it's an unstable fixed point because they have positive growth rates. Okay. So what that means is that here is a fixed point, but it's unstable. We can draw it as a circle if we like. And actually, it's going to come out up here in both directions. Because indeed, we know that n1 on its own is going to grow up to its carrying capacity, and 2 on its own is going to grow up on its carrying capacity. And indeed, all the trajectories are going to be leaving the origin there. All right, so the trajectories are coming out. But then once they get to the line, they have to be going vertical. Right? Coming through here. And indeed, okay, so in this point here's a K2. Yeah, so what's going to happen is that they look like this. All right, so they kind of come out. Once they hit the line, they go straight. If you started out here, then they might come in, and that might look like the following. Okay. So this is what the trajectories look like over time. All right, so given this, we want to make sure we're on the same page. Okay, what's the outcome of the competition in this situation? All right, 1, 2, 3, 4. You can vote 5 if you're unhappy. All right, ready? 3, 2, 1. All right. The people that are not voting, is it because but then you should vote 5 to indicate your unhappiness. No? Okay. All right, yeah, so indeed, species 2 is winning. All right, because what you see is that it doesn't matter where you start, all these trajectories go to this location here, where species 2 is present at abundance K2, species 1 is present at abundance 0. Right? So this is the situation corresponding to species 2 wins. All right, species 2 drives species 1 extinct. Okay. Could species 1 have survived on its own in the absence of species 2? Sure. Right? So this is a case of competitive exclusion. Right? You can imagine species 1 on an island happily surviving at carrying capacity 1. Right? Then an invasive species comes, species 2. Right? What it does is it drives it extinct, and you can see what the trajectories are going to look like, where they look like this. So this is the trajectory corresponding to starting out species 1 on its own, small number of species 2 invaders, and drive species 1 extinct. Okay? All right, so given that, what do you think is going to be the outcome of this one? All right, 10 seconds, and then we're going to vote over here. Ready? 3, 2, 1. Right? Species 1 is going to win, right? Because this is kind of the flip situation. And one thing that you can come back and look at this and ask, all right, well, when is it that you think that species 2 should win? Well, species 2 should win if this beta 1,2 is really big, right? Because species 2, if it's strongly inhibiting species 1, then, yeah, that's when it should win. Indeed, we can imagine, okay, so beta 1,2 being big, that corresponds to pulling this line down, right? So that makes sense. If you have a strong inhibition of species 1 by 2, you pull that line down, and that's where species 2 is going to win. Okay. All right. This one's a little bit trickier, so let's stare at it for a moment. All right, do you need more time to vote on this one? Maybe another 10 seconds. Oh, you're talking about right here. Right, so, okay, so what does it mean when those two lines cross? Right, it means that it's crossing the isocline for both N1 and N2. Right, so this is the definition of a fixed point. Because dN1T and dN2T is equal to 0. Of course, depending on the stability of that fixed point, it could change the situation, yeah. Okay, let's go ahead and vote just so that I know where we are. Ready? 3, 2, 1. Right, so this one's a bit harder, I would say. All right, so broadly, I think that there's an opinion that it's going to be either 3 or 4, probably, right? Okay, why don't you, let's spend the 30 seconds, turn to your neighbor and discuss, because I think it's worth the time. Okay, well, why don't we go ahead and draw some of these things out? Okay. All right, now, in this situation, again, is the origin still going to be an unstable fixed point? Yeah, sure. Okay, so it's still unstable. We haven't changed anything. We're just changing the... Oh, I'm sorry. Okay, over here. Good. All right. Now, all right, what's going to be the orientation of the trajectories on the solid line again? Still vertical, right? And over here, it's going to be horizontal. Okay. So now imagine you have trajectories leaving the origin. What are they going to have to do? Well, this trajectory, it's going to come and crosses here, and it's going to be forced over to the left by this guy. So it kind of comes up like that. Some other trajectories here might behave rather differently. Okay. And this is one that kind of curves over. And right. So what you see is that in this is the situation where the trajectories get pushed away from each other. And that's because this, if you check, this is also an unstable fixed point. So it's a fixed point, but it's unstable. It's not attracting. And similarly, there are going to be trajectories coming out from outside. Right. So here, there's going to be some separatrix. There's going to be some, we'll say a dotted line, that is separating the two basins of attraction. Okay. If you start out basically above and to the left, you come over here to this stable fixed point. But if you start out anywhere here, you come over here to that stable fixed point. So this is the situation where you get bistability. You can go ahead and just draw this over here because it's fun. All right, so this is vertical. These ones are horizontal. Okay, and the trajectories here now are going to get pushed inward. So you have something that looks like the following. Okay, so everything comes together because this here is now a stable fixed point. And I want to make sure I get the right one. Solid. Okay, so yeah, this one again is K1. That's K2. Okay. So here is coexistence. Okay. All right. Okay, so we can summarize what happens in a very simple way. You can define these alphas that are kind of the normalized interaction coefficients. They're normalized by the ratios of the carrying capacities. But often, I think, to try to simplify things, it's useful to just, you can also imagine it just, well, what if the carrying capacities were equal? In that case, this would be 1, and the alphas are just equal to the betas. Okay, so when the carrying capacities are the same, there's a very simple statement, which is that depending upon whether the alphas, and so if we plot log, okay, now I always forget which one I want to do. All right, so let's do 2 wins, 1 wins. All right, so this is a log of, okay, so species 1 wins if it has, if beta 2, 1 is large. So this is alpha 2, 1. This is log alpha 1, 2. Okay. All right, so the outcome within this model is rather simple. In particular, if you just set the carrying capacities equal, then it's just about whether each species inhibits its opponent more or less strongly than it inhibits itself, right? Because log of alpha here, that's telling you if log alpha is greater than less than 0, that's the same thing here as beta being larger than smaller than 1 when the carrying capacities are equal. Okay? All right, so if you have the same carrying capacity, then basically whether you survive or not is just dependent upon whether your opponent is inhibiting you more or less strongly than you're inhibiting yourself over here. If they both inhibit each other quite strongly, you get bistability. If they inhibit each other weakly, you get coexistence. And then each species driving the other one extinct depends on whether they inhibit their opponent more strongly or not. Question? Yeah. Sure. Well, we start at 0, and we know this is unstable. So given that we know this is unstable, all these arrows have to be pointing out. Right. Now, they're coming out, and they have to become vertical, coming here. So that already specifies the direction of the arrows. Oh, I see. You're saying here, okay, because you're saying that maybe they could curve out. Okay, well, all right, but the other thing that we know is that if you start out at infinity, everything's coming in, right? Because out at infinity, there's strong inhibition, so that means all the trajectories have to be coming in from out there, so then we know they're coming. So here it kind of goes, is that? Yeah, so we can do the intersection ones. All right, well, they're coming in from out here, right? And that means that, well, certainly when they cross here, they have to be coming down, right? And when they cross here, they have to be going to the left, right? So that means at least we know the arrows are here. Similarly, we knew the arrows were coming out of here, and they have to curve around to cross. So in that sense, we know that they were coming in at this location, right? And that's, I think, sufficient to, it might not be enough for a mathematician to be convinced, but all right. Any questions about where we are on this? Yes. Yeah, so you should definitely calculate the stability of the fixed points. It's just that that takes more time, and it's a lot of algebra up on the board. Yeah, so I guess, yeah, but if you wanted to analyze this, I would say I would do the isoclines, I would do the trajectories, and then to really convince yourself, then I would do the stability of the fixed points. But I think everything I said is true if you go and do the fixed point, the stability analysis. But yeah, it's just that, there gets to be a bit more algebra when you, but you should go and calculate the Jacobian and the eigenvalues and. Yes. Yeah. Right, the question is, how do we know the trajectories are coming in when we start out in infinity? Well, so long as the betas are positive, right, so long as it's a model of competitive interactions, then if n1 and n2 are both going to infinity, then we know that this thing is going to become larger than 1. Right, so we get a negative growth rates. Yeah, there are lots of ways that we could, yeah, so what you're saying is that we could actually go and confirm where is it positive, where is it negative. Yeah, I agree, because everything below, yes, that's right. We could figure out where it, yeah, and that's another way of getting to it. All right, so, yes. Yeah, the lines coming out of the origin do not actually, that's right. I agree with you, although I also am not sure how I would justify the statement that they're not going to cross the dashed line. So I'm hesitant, it's a true statement, but I don't know how to argue it. So I don't want to invoke it, is all. Oh yeah, no, that's true that the trajectories can never cross, right? Because here what we have is a two-dimensional dynamical system. It's a first-order differential equation, right? So at any given value of n1, n2, there's a unique direction of dn1, dn2, right? So none of these trajectories can cross, and and I've tried to avoid doing it when I drew it, but yeah, the trajectories cannot cross. So that's also why, for example, you can't get chaos in a two-dimensional first-order differential equation, right? Because they can't cross. Well, I think that the reason they cannot cross is because you get a unique value. If you plug in n1 and n2 into these equations, you get an answer, right? If you have crossing of trajectories, that's two answers, right? Because a crossing of a trajectory would mean that at that point, you could have two different arrows, right? And that's not consistent with the statement that plugging in an n1 and n2 gives you an answer, right? Yeah. All right, so I want to just discuss one more thing about this model, and then we'll switch. In particular, I want to think about this question of whether it would be possible for us to use this model to describe a mutualism. All right, so if you wanted to use this to study a mutualism or to model a mutualism, how would it work, or what would you do? Would somebody like to suggest what we would do for a mutualism? Betas could be negative. Yeah, let's try it. All right. All right, so we're going to now just look at the isoclines if you put in negative betas. All right, so first of all, this solid isocline for dn1 dt equal to 0, that should still start at K1, right? Because in the absence of species 2, this is where the abundance is going to go, right? But now if I have a negative beta, what's going to be the direction of the line leaving this point K1? All right, so let's think about it for a moment, and then you guys can help me by pointing. You guys understand the question? I want to draw the isocline for species 1 if we have a negative beta. And we're going to assume that both the betas are negative, but all right, so we're going to assume that we started out here. Question is, does the new beta, is it going to go like this, that? I don't know. Okay, so elbow fixed at K1. You can tell me where to draw it. Okay? Ready? 3, 2, 1. All right, looking good. Yeah. in this case, if you have a negative beta, it means this line is coming, it's positive slope now. Right? Because it's saying that if you add species 2, now the condition, you can actually have more N1 before your dN1 dt is equal to 0. Right? And you get the same thing is true for species 2 if that beta is positive, or sorry, if that beta is negative. Okay. This is what the isoclines look like for a mutualism. All right, so that's beta 1, 2 and beta 2, 1, both being less than 0. Okay. And do you think that this is going to be a stable or an unstable fixed point? All right, you can verbal answer, stable or unstable. Ready, 3, 2, 1, stable. Yeah, so this one's indeed going to be a stable fixed point. And all these things are unstable. Okay. So the trajectories are going to leave. Again, it's going to be vertical here, horizontal here. So if you start out in here, then you always, you can never leave this section. Right? You may curve out a little bit, but so it might look something like. But then if you start out, this one kind of comes here. And if you start out here, you end up with a trajectory that looks kind of like this. Coming together. Right. The key thing is that at equilibrium, this is the N1 star or whatnot, at equilibrium, both species have an abundance that's larger than what they would have had on their own. Okay? So that's the sense in which it's a mutualism here. One thing that's worth noting, and we're not really going to talk about mutualisms for today, but this is a model of what you might call a facultative mutualism. Right? They're benefiting each other, but they do not require each other. Right? You can see that either species could have survived on its own. It's benefited from its partner, but it's not required. Okay? So this Lotka-Volterra interspecies model cannot be used to capture the dynamics of an obligatory mutualism, where the species need each other for survival. All right, if you try to do it, what happens is that the species abundances go to infinity. All right, so in that sense, you could, it sort of is an obligatory mutualism in the sense that they couldn't survive on their own, and then they do survive together, but it's not a very satisfactory model. Right? Yes? Oh, the facultative, or sorry, the obligatory mutualism stuff that I was just talking about. Yeah, so for that, what you would want to do is, it's like you put in maybe negative R's and negative K's, and so then the solid line would have crossed out there. Oh yeah, so it looks like this, where now this is a stable fixed point, this is unstable, and then it goes out to infinity. All right, so this would be an attempt at an obligatory mutualism. And again, solid line is, yeah. So here the trajectories are coming into extinction. And that's true out here. This wasn't very horizontal, sorry. So what you see is that there's going to be some separatrix. In here, everything goes extinct. Above it, everything survives. But it survives by going to infinity. Right. So if you want to model an obligatory mutualism, you should use some other model. You need some way of getting these isoclines to turn around. Fundamentally, the problem is these isoclines are lines, so they can only cross once. But what you really want is to have curves that are curvy, so they can cross again, and then you get a stable fixed point out here. Okay? Yeah. That's right. Yeah, so the issue here is that to do this, you have a negative K1. Yeah. I just wanted to mention that in case anybody is interested in modeling mutualisms, that this model can be used to model a weak or facultative mutualism, but not an obligatory mutualism. All right, with that, let's switch to the PowerPoint. Okay, so general motivating features, right? So my group is broadly interested in using experimentally tractable microbial communities in the lab in order to explore the kinds of ideas that we've been talking about throughout the week. Right? So these kind of general ideas that we find exciting within evolution and ecology. And so it's partly to probe these fundamental principles, but partly we'd like to understand microbial communities themselves. Right? The motivation for the microbial community piece is, of course, that over the last decade, it's become increasingly acknowledged that microbial communities are important in a wide variety of different ways. So, for example, the recent results encouraged The Economist to make this kind of hyperbolic statement that is obviously not true, but it's more true than we thought 10 or 15 years ago. It's, I think, though, important to note that although it's maybe only recently been acknowledged how important microbes are for human health, we've known for a long time that microbes are essential for the health of the planet. For example, something like half of all the carbon cycling that takes place on the planet is due to microbial activity. Okay? So we'd like to understand how these microbial communities are working, but a major challenge is just the tremendous complexity of these communities. All right, so this is an electron micrograph of human feces, and what you can see is that it's complicated. Right? Well, and not only that, there are a lot of microbes there. Actually, a majority of the dry weight of feces is microbes, which is amazing, right? And so it's been false colored, but you can see that there's a lot of different kinds of microorganisms, different shapes, sizes, and so forth. Okay? These communities are tremendously complex. Okay? So the question that maybe pervades much of ecology and actually evolution is just to try to understand the origin of the tremendous diversity that we see in nature, right? In the context of ecology, how is it that so many different species can coexist within, say, the human gut, or the soil, or elsewhere? Now, there are many different possible explanations for this, but one set of explanations that have gotten a lot of attention recently is what you might call kind of multi-species or higher order kinds of interactions. Right? So it could be, for example, that there are many of these rock, paper, scissors kinds of dynamics going on, and that could stabilize species within a community because no species could become very abundant. I'm not going to say anything about this today. We've been searching for these things within natural communities. We've looked at thousands of different trios, and we can't find any examples of them. So our current take is that rock, paper, scissors trios are actually not a significant contributor to at least microbial ecological diversity. Okay? If you're curious about such things, you can ask me later, because I'm not going to say anything more about it. But another kind of higher order interaction that people have been thinking about is this question of how often is it the case that a third species fundamentally changes the nature of the interaction between two other species. Because if these higher order interactions are ubiquitous and strong, then they could be a significant stabilizing force for the diversity within these complex communities. So one of the things that we in my group have been very interested in is just asking this question of, to what degree do pairwise experimental measurements allow us to predict the outcome of multi-species competition? All right, so just so you have some sense of where we're going, so we're going to start by just asking the question, well, what's the typical outcome if you compete a pair of species? So you pick random pair of species, put them in some environment, like a pyramid and under the ice in Antarctica. All right? The question is, well, what's the typical thing that happens? All right? And then, based on those pairwise outcomes, we're asking, can we use the experimentally measured pairwise competitive outcome in order to predict what happens when we have 3 or 4 or 5 species present? Right? And the answer is that typically we can. Okay? And finally, we've recently been thinking about how is it that these communities change under stress as you go from, say, a healthy environment to a challenging environment. And it turns out that there are some very simple rules by which these pairwise competitive outcomes shift. And given that the pairwise competitive outcomes can typically be used to predict the outcome of multi-species communities, that means that we can really go from very general, simple statements to predicting how complex, well, moderately complex communities change under stress. Yes? Yes. Yeah, so our claim is that pairwise interactions are the dominant effect. Yeah. Not everybody agrees with this, but what can you do? Right? All right. Okay, so just to be clear about what our experiments are going to be looking like, they're very simple. We take a pair of species, we're competing them in liquid culture, excuse me, either well mixed or not. It actually doesn't make very much of a difference for all the stuff that I'm going to be talking about today. We allow them to grow and compete, use those resources, and at the end of the day, we're just going to perform, again, this dilution, like I told you about before, where we transfer a small fraction of the organisms into fresh media, and we repeat this process. We typically go for about 60 generations, with the idea is that we want to reach an ecological equilibrium before there's time for significant new mutants to arise in the population. So we're really trying to focus on ecology before we try to tackle evolution in these contexts. There are two of you guys. Yeah, maybe you first. All right, so the question is whether the assumption of pairwise interactions being dominant also holds for long-range interactions. And what I want to stress is that I'm not a theorist, so I don't make assumptions. I just go and I make measurements. So my claim is that experimentally, the pairwise interactions are dominant. Okay. All right, and then the question is long-range interactions, right? So most of this stuff is in well-mixed environments, so it's not, although we've done both, I'm going to say I think the answer is yes, that I think that the pairwise are dominant even for long-range interactions. Oh, you're wondering about the different contributions of short and long range. Yeah, well, I'd say for now, you can think about it as all being well mixed, so there's no such thing as short versus long range. It's just that everything is going to some pool that is all being shared. But I think it's an important question, and it could end up being different, although I think it ends up not being, but it's an important question, though. Yeah. Yeah, indeed. Yeah, so we're picking a bunch of random species. They could be mutualistic. What we see is that most of the interactions are competitive, and some of them, in some cases, a species benefits from another species, but in almost no cases do we see a two-way mutualism based on these random species being combined. Yeah. So it's mostly competitive. Yeah. All right. And in this starting experiment, what we did is we just took 8 different soil bacteria, 6 of them being Pseudomonads, an Enterobacter, and a Serratia. Here, E. coli is just shown as an outgroup. Right, so what we're going to do is we're going to compete all these species in pairs, and then we're going to ask, from those pairwise outcomes, can we predict what happens in all of the trio competitions? Okay, so can we use pairs to predict trios? This is work that was led by Jonathan Friedman, who was a wonderfully talented postdoc in my group and is now starting his own group at the Hebrew University of Jerusalem. He is looking for students and postdocs, so I encourage anybody to think about it. Okay. All right. So maybe the first thing to say is that among these 8 species, we see essentially 2 simple outcomes. Okay. All right. Some fraction of the time, what we see is competitive exclusion, where it doesn't matter where we start the competition, what we see is that one species, in this case PP, drives the other species, in this case PV, extinct. All right? And what we're going to do is we're going to visualize this just by an arrow that goes from the loser to the winner, because this is like the flow lines of these kinds of trajectories that we're going to be talking about. The other thing that we often observe, and actually in these 8 species, this is the most common outcome, is stable coexistence. Here again, you can see that it doesn't matter where we start the competition. All of these simple communities go to the same outcome, where the 2 species are coexisting at a stable equilibrium fraction. Right? We're going to denote that by these 2 blue arrows coming together to that equilibrium. Okay? Yes. Yeah. So the phylogenetic relationship in this experiment there's no signal, right? So the closely related versus farly related, further related species, more distantly related species. You wouldn't think that I'm a native English speaker, right? But yes, so it does. Yeah, so the probability of coexistence does not depend upon how closely related the species are. Same for, and here, these are the only two outcomes, so yes. And we designed a larger set of species in order to try to get at the question that you're talking about right now. And because as a physicist, I love the idea that a bunch of things don't matter, right? So I love the idea that I don't even need to know what the species are, or where, and because it doesn't tell you any information. So we don't, there's a lot of debate actually within the field regarding how the phylogenetic distance should influence the strength of positive and negative interactions, because you can come up with good verbal arguments for why it should go in either direction. But for now, you can just think of these as letters, because it doesn't have any correlation. Okay, sorry. So the first letter means that it's a pseudomonid, and then the second letter is telling you which of the because there are 6 species that are that are pseudomonids. So so the 2-letter combination is just telling you which species it is. All right. So then we go and we do a bunch of measurements, right? And after we do that, this is what the network, and this is the experimentally measured network of pairwise competitive outcomes looks like for these 8 species. And remember, the arrows go from the loser to the winner, right? So for example, here what you can see is that this PP is perhaps the most fit of those species, because it can outcompete 3 others and it coexists with the other 4. Right? Whereas some other species lose to a bunch of this, right? So you can kind of look at this, stare at it, and get a sense. You can look at the network motifs, like what we were hearing about this morning. In this network, we also don't see any rock, paper, scissors trios, and that's part of what motivated us to go look at a 20-species community isolated from a single grain of soil, where there, again, we can look at over 1,000 trios. We don't see any rock, paper, scissors trios. Okay? But today what I want to do is tell you about this other question, which is, if I tell you the pairwise outcomes, can you predict what's going to happen when we mix 3 species together? Yes? Definitely. Yeah, so all of these experiments are done in a particular environment. It's a minimal media with 2 different carbon sources, serine and galacturonic acid, which again don't mean anything to me. They probably don't mean anything to you. Yeah, so indeed, all of this will change if you change the temperature or the environment of it. And we're interested in all of those things, and we're chugging along. Yeah, but for now, this is all of this is just in a single environment. Yeah. So what we're doing is we're measuring the pairwise competitive outcomes in an environment, and we're trying to predict the trio outcome in the same environment. Because of course, you can always change the environment and things. Yeah. Okay, so then what we want to know is, okay, well, what happens in trio competition? Oh yes, and we have our alien v predator. All right, who's seen the movie? You guys. They even have a movie night here. I should have arranged a Friday night movie showing because I think they do have Friday night thing. We should have. That would have been a great introduction to the ideas of it could have been a required movie. Okay. All right. Well, if you're going to watch it, you can cover your ears because I'm going to give you a spoiler. The basic idea is it combines 2 wonderful series, the Alien series and the Predator series, and puts them into one amazing movie. Where the question is, there's a long history apparently of the Predators coming and training, testing their battle skills against the aliens, and it's in this underground pyramid, under the ice of Antarctica. It's great, okay? But then, and it turns out that the Predator, as everyone knows, is more powerful than the alien, partly because of this shoulder-mounted laser beam that he has. Okay? But in the movie, there's a third species that enters, and they mess everything up because they actually kind of accidentally get this laser weapon before the predator can get to it, and then it makes it for a more complex 3-species outcome, right? And so it's not obvious what happens when you compete the 3 species together. Right? So this is what we're trying to do in our lab, is compete 3 species together and ask, can we predict what's going to happen based on what we know about what happens in the pairwise competitions? Okay? For sure. Exactly. Yeah. So the statement here is that, this is counterintuitive because the pairwise outcomes clearly don't have all the information of what could be happening when you have the 3. So there's no reason that it has to work, right? And importantly, I think it's an experimental question because there certainly are higher order interactions. In biology, and there are 3-way, 4-way. In principle, there are infinite way interactions. So it could be that it's totally hopeless. Right? So for me, it's an experimental question of whether we can say anything about the trios based on the pairs. Yeah. All right, so here's kind of a random trio. Right? And I want to remind us what these arrows mean. Right? What this is telling us is that the Serratia is outcompeted by the Enterobacter. Right? So SM loses to EA, but the other 2 pairs display coexistence. So this is experimental measurement. Right? Now what we want to know is, if we compete all 3 together, what should happen? And this is worth you should think about it before we do the experiment, just because it's good to do. Right? So if this is the only information that you have, right? Of course, I could give you the genome sequences of the species if you think that's going to help. No, but in practice, the question is, if you only have this information, what do you think should happen? What's that? Well, I don't know. So what I'm showing right now is just the pairwise outcomes. So I haven't shown you what happens when the 3 species come together. And I will, but I think I just want to first encourage you to and one thing that's actually nice about this framework here, it's a triangle that you could just view as a triangle that shows the pairwise outcomes. But you could also view this triangle as the simplex corresponding to the relative abundance of each of the 3 species. Okay? So anywhere in the interior corresponds to the 3 species being present. Right in the middle is an equal abundance of the 3. And as you get closer to each of the species names, that corresponds to more of that species being present. So for example, this point up here at EA, that would be all of EA and none of the others. This point here, where it's coexisting, okay, it means it's coexisting between the 2, and EA is extinct, and vice versa. Right, so any point on this triangle is telling us about some relative abundance of each of the 3 species. All right, so what we're going to do is vote, because we like to point places. And the question is, if we do an experiment where we start at different locations on this triangle, different relative abundances of the 3 species, what do you think is going to be the final outcome of the competition? So, are you ready to point in some direction, or do you want to think about it for? All right, we're going to point. All right, ready? 3, 2, 1. All right, but don't point at the screen. Point somewhere here. So this would be EA winning. This would be PA winning. That would be SM winning. Or you can do coexistence by doing somewhere in between. All right, so somewhere over here, maybe. All right, yeah, I think that's fair. All right, and we'll talk about where maybe that intuition came from and da-da. But of course, the point of making predictions is that you want to go test them, right? So what we did in this case is we started with replicate communities, 2 over here dominated by SM, 2 here dominated by EA, and 2 here dominated by PA. And then we compete them for the 60 generations, and we just measure the species abundances over time. And when you do that experiment, what you get is this. Okay? All right, so we started at different places, and relatively quickly, all of those 6 replicate communities all go to the same place over here, where SM goes extinct, and we're left with coexistence of EA and PA. What you can see is that, see that this was what we had measured before for the 2 species and the 6, all right, so that means that those 6 replicate communities go to the same place, the same fraction as we had measured before in an independent experiment. Right? So that tells us that it's well behaved, right? And all 6 communities go to the same place, and there's and this is actually just measurement error as well. Because of course there's going to be some of that, right? So this is saying that indeed there's that maybe the simple intuition that many of us had coming in here would say that, oh, since SM loses to EA in pairwise competition, then maybe it's also going to be driven extinct in the context of the multi-species community. And indeed, you could kind of capture that, and that would leave with just these 2 coexisting. All right, so then you can just have a very simple community assembly rule to say that we're going to predict that a species is going to survive in the multi-species competition if and only if it can survive against all of its competitors in pairwise competition. All right, so the goal, and then for more complex communities, the goal is to find what community or one or more community states would satisfy this thing where nobody is excluded by anybody else in pairwise competition. Okay. Yes? That's right. So in this case, what we were actually doing is every other day we were diluting by 1,500, and then we went for 6 cycles. Yeah, so it might be 70 generations. Yeah. So here, at the end of each cycle, they reach the same. They reach really pretty much a constant population size, and that I think it does depend on how long you're diluting by and so forth. Yeah. So here, actually, I'd say the fractions are pretty well behaved, and that's why you can just put it onto a simplex and nothing funny happens. Yeah. No, so we think here that this stable fixed point is really right on the edge where SM is extinct. Yeah. Despite the concentration of... Yeah, right. So in this case, we started at many different locations here, and the question is, where do they all go? And the statement is that all of these trajectories go to this fixed point over here. In some ways, that fixed point of coexistence between the pair, that's a stable fixed point, and this other one ends up being an unstable fixed point. Yeah. Of course, this is just a trio, and I think that you can kind of be convinced that this is what happens in this trio, right? Because it happens in all the replicates and so forth, but it's just one trio, and in principle, anything could happen in any given trio. So what we need is some sort of statistical information regarding how frequently does a given kind of assembly rule or whatnot actually work. Okay? So then what we do is we go and we make lots of measurements. Right? So there are choose 3 trios, and we go and we do all of them. For all of them, we start in those different locations like I showed you before and look to see what happens. Okay? And the basic statement is that with 90% accuracy, that simple assembly rule that I told you about will predict the survival of a given species in the trio. All right, the 10% of the time, roughly, where it fails, it is roughly equally divided between false survival and false extinction. All right, so typically, the pairwise competitive outcomes work for predicting survival in a trio, which to us tells us that these higher order interactions are not dominant, at least. There's something else that's very much worth saying here, is that the Lotka-Volterra model is one way to capture interspecies interactions, and it will make some set of predictions going from pairs to trios, but there are typically multiple allowed outcomes. For a given triangle, there are multiple different things that could happen. Okay? So from that standpoint, even the failures of this simple assembly rule do not have to be caused by higher order interactions. They could just be caused by, even because the Lotka-Volterra model is a purely pairwise model, but it still can have cases that fail. Okay? Yes. Yeah, right. Yes, so indeed, no, because even if you have a minus, and I'll talk about this more, but even if you add a minus delta, you can reparameterize into a new version of, yeah. Yes. Yeah. Yeah, so the question is whether we could parameterize the logical Volterra model. And you can always do that. And the statement is that that makes worse predictions than this simple qualitative rule that I just told you guys about. And there could be multiple reasons for that. First of all, the species are not actually logical Volterra species. They're not saturating logistically, like we mentioned on Monday. They're not interacting in this very simple way that's assumed here. And it may be that maybe some of the outcomes are just too rapid, so we're not parameterizing it very well. But the empirical observation is that trying to parameterize this model, does a worse job than just this simple rule. Yeah. Well, you can always parameterize it, and you have enough free parameters that it looks pretty good. But then you have to decide what that means. Right? Yeah. Right. Yep. Right. That does not work. Exactly. So if you parameterize based on the pairs, and then from that you make predictions for 3, 4, whatever, that does not work. And in particular, it does worse than this very simple thing. Yeah. Well, no, we didn't start. I mean, we started in all 3 corners. Well, it ended up near here. Well, if you start at 3 places, then the final state will always be kind of close to one of them, I think. So I don't think it's anything more than that. Well, I guess this is what I'm saying, is that since we start at multiple locations on the triangle, then you can't be too far away from one of those starting locations just because you're starting multiple. So I think it's hard to answer. Yeah. No, but we really believe that for most of these cases, we could start, like in this case, we could start anywhere on the triangle and we would come to that. So in this case, we think there's a single, unique, global, stable, fixed point. In some other cases, we do see bistability, but it's very rare. Okay, I do want to show an example of another triangle where this simple assembly rule fails. What you can see is there are 3 different species now, shown here. The outer edges look the same as the previous triangle that I showed you, where on the left we have a case of exclusion, the other 2 display coexistence. But now what you can see is that there's a bit of a surprise in terms of the final outcome. Because here it's the species that is most fit in the context of pairwise interaction that is actually the species that goes extinct in the trio. And it's not just that, it's not just experimental noise because you can see all 6 of these replicate communities are coming here. And indeed, you can even see that the trajectories are curving around as it goes to here. So this really is like the outcome when you compete these 3 species, right? It's a failure of our simple assembly rule because we're surprised that PP up here goes extinct, but it's definitely the outcome. So we're reliably wrong, and that's good. That it's in the sense that you want to at least have reproducible measurements so that, even if it's a surprise, at least you know that you're actually wrong, right? The worst thing is when, the world is just like, oh, you might be right, you might be wrong. Whereas here, okay, yeah, we're definitely wrong. Okay, but that could point us towards interesting things from a dynamical perspective, from a molecular interaction perspective. We could search for possible higher order interactions, right? Because it could be that, for example, PCH could be breaking down a toxin that PP is making to kill PV. That's a story that could explain this trio. We have experimental data that suggests that's not what's going on here. But I also want to highlight that it doesn't have to be a higher order interaction, because this is one of the allowed outcomes within the Lotka-Volterra model, even though the Lotka-Volterra model is a purely pairwise model. Okay? So this is allowed from purely pairwise interactions, despite the fact that it looks quite surprising. Yes? I'm not going to say very much about 3 to 4. In general, as you get to larger numbers, predictability eventually is going to get worse. Although we did recently show that in our 20 species community, where we measured all the pairwise interactions, these assembly rule actually did predict the exact 3 species that would survive when you mix all 20 species together. But that's, it's one data point. But the data point has 20 things. So, yeah, you can argue about what it means. Yeah. All right, so I only have a few minutes. I'm just going to give you a brief sense of what we're doing right now in this regard, because I think it's fun and it relates to what I told you about before. And this relates to this question that Sid asked. Okay, I just want to mention, we also do sometimes see bistability. Okay? So that is a relatively common outcome. These are the 3 things that we see for 95% of pairs. Okay, so that's good, because this is what happens, or these are the basic outcomes in the Lotka-Volterra model. So even if it's not a perfect model to describe microbial interactions, it still gives you some basic sense of what's going on. I do want to mention, what happens? All right, I already told you about this model. The question is, what happens if you add a mortality rate? So this is what Sid was asking. Okay, and this you could think of as just some sort of environmental deterioration that is harming both species in the same way. They experience the same delta. Okay, as I mentioned to Sid, you can just reparameterize these things, pull the delta in there, and the great thing is that in this model, it makes a very simple prediction, which is that you could start out here with the slow grower winning, because it's a strong competitor. It strongly inhibits its partner. And as you increase the mortality rate, you go down to the right at a 45-degree line on this log alpha scale. All right, so the expectation is that you should eventually be able to get the fast grower to win. But along the way, you should cross a region of either coexistence or bistability, but not both. Okay? And what's wonderful, I'm going to skip over the data, but this is true in the sense that we can go and do experiments, we can increase the dilution, and we reliably march across this thing. We can cross coexistence or bistability. So, and this is really, and the nice thing is that this is a robust prediction, and it's really even broader than just this model, because again, this comes down to this is like a bifurcation analysis. So any sort of well-behaved model there's a limited number of things that any sort of well-behaved dynamical system can do, right? So in that sense, it's maybe not surprising that this kind of prediction works, but on the other hand, it's nice that it works. And then I'll just mention that we can then use, I'm not going to show you the data, but given that we now have, we've already shown that pairwise outcomes typically predict what happens in, say, 3 species, and it kind of works for 4 or 5, okay? And now we have a sense of how the pairwise outcomes shift as you change the environment. Okay? For example, here, the slow grower wins, then there's a region of coexistence, and then the fast grower winning, right? So now we have some sense of how the pairwise outcomes shift. Now we can combine those things to make predictions for how communities are going to shift as you vary the environment, right? And this also works. Not only do these community assembly rules work in vitro, in test tubes, but we've also now demonstrated they work in the gut of the worm. So we took 11 different species, did all the pairwise competitions in the gut, and then we went and did 20 or 30 different trios. And again, the pairwise assembly rules do a pretty good job. Okay? With that, I'll quit. So, so we're excited about these pairwise rules because they're interesting objects, and they actually do a pretty good job of predicting what happens in trios, although this is a trio that is pretty, but where it fails. Okay? We also have some sense of how the pairwise outcomes shift as the environment changes. And also, we can combine these two statements to predict how simple multi-species communities shift as the environment shifts. Okay? And with that, I'll thank the group again. And I think we're out of questions, or well, we might be out of questions, but maybe one or two? All right. Yeah.
Okay, thanks. So you didn't show, but I wanted to ask what happens instead when you have trios in which basically all three pairs would coexist?
Mm-hmm. That's a good question. All right, so the community assembly rule would predict that all three would coexist. And what we see empirically is that that's true only a slight majority of the time. The other significant minority of the time, what we see is that one of the three species goes extinct, and then we're left with coexistence of the other two.
In that case, can you conclude like that essentially what you somehow that the interaction between the three species, or let's say that your dynamics is not essentially the kind of replicator dynamics or the evolutionary dynamic that you would get?
I think we cannot conclude that because, again, also in that trio case that you're talking about, in a logical Volterra model, if you have three pairs that coexist, then the two allowed outcomes are for all three to coexist, or to have one of them go extinct to have two coexist. And indeed, for all of the trios that we have, basically all of them are one of the allowed outcomes in a logical Volterra model. So from that standpoint, I'd say we have very weak evidence for any sort of higher order interaction, because basically everything in principle could have happened in a logical Volterra model. That doesn't mean that it is a logical Volterra model, but it means that, it's not that we have an outcome that's like, oh, this couldn't possibly happen in a logical Volterra. Is that answer? Yeah. Or maybe a last question, and then we can have lunch.
So in the worm, how do you control the gut environment? So in the lab, it's possible.
Yeah, the question is, in the worm, how do we control the gut environment? Okay, so there are, I think, two or at least answers. One is that part of the appeal of the worm gut as an environment is that we're not controlling the environment, is that the worm is controlling the environment, and so it's a more natural environment. So that's nice because then it's biological relevance or something, right? Okay, but of course, the reason that we're using the worm is because we actually can control it, right? So so in particular, we have, we can take advantage of, the last 30 years of worm biology, where there are mutants that have enhanced immune function, poor immune function, that have impaired gut peristalsis, that have impaired munching of the bacteria. So we can actually control a bunch of things in the gut, and this is the kind of thing that we're going after. Yeah. I think Sid is going to decide whether we quit. Okay. Yes, exactly.
So that is the helping thing.
Yeah. So C. elegans, what they do is how they eat is they eat bacteria, and that's great because then we just take the worms, we put them in an environment with some bacteria, and they eat. In our first paper from the worm work, we demonstrated that the initial colonization event was a rare event, and that could drive tremendous diversity or heterogeneity between different gut communities, depending upon which colonist happened to get started first. But there's a lot of things going on. Right? All right. Thank you. Yeah.